It's a basic human need to ask, who are we? Where do we come from? How did we get here? Why do we look the way we do?
The story of our evolution is just a small chapter in a much larger story. The evolution of all living things.
Evolution shows us that we're much more connected to the rest of the world, the rest of animal life, than we could ever have imagined.
We can recognize the connection to our closest relatives, but when we know how to look, we can also find it in other mammals, birds, reptiles, fish, even insects. The search for the genetic mechanism of evolution took most of this century. When scientists finally found it, they were astonished by just how simple it was. One of the key players was Mike Levine. One insect in particular became an object of fascination.
They have a quick generation time, and they have lots of pattern. I mean, you wouldn't know it if you look at a distance, but when you look under a microscope at an adult fruit fly, you'd be astounded by the number of bristles, the intricacies of their wings, the patterns of their eyes. But the embryos are something else. I do love the embryos.
Scientists had long suspected that embryos held clues to how animals evolve. All embryos start out as clusters of nearly identical cells. But soon, an embryo partitions itself into specialized segments, which develop into the final form of the animal. What controlled this process? How did the embryos know what shape to take? One of the first people to study these questions was a 19th century naturalist named William Bateson. He also observed that animals occasionally developed with some segments in the wrong places.
Insects with legs in the wrong place, crabs where a claw was transformed into a leg.
To Bateson, these developmental errors meant that the underlying blueprint for the animal was being disrupted. He had no idea how it happened, but he suspected that these random changes might provide the fuel for evolution. By the 1940s, scientists working with fruit flies had learned how to cause disruptions in the developmental blueprint by dousing growing embryos with radiation and poison.
And so when they did that, they found flies with changed wing structures, changed legs, and these very special flies that have one part of the body in the wrong place, or a copy of a normal part of the body in another place.
The scientists had triggered the changes by damaging the fly's DNA. Within each cell of the developing embryo is a chain-like molecule called DNA. The experiments showed that DNA was somehow causing the embryo to divide into segments. But how? Scientists were just beginning to grasp that the DNA itself was made up of segments, called genes. The question was, how did the genes shape the body? One researcher, Dr. Ed Lewis of Caltech, studied this question for 30 years by crossbreeding thousands of flies. Lewis's work led him to a controversial idea. He proposed that a surprisingly simple mechanism was shaping embryos. He wrote that each segment of the fly was being directed to grow by a single gene. A small set of genes, a kind of genetic toolkit, appeared to be laying out the entire body.
And as you looked at these genes, he said, this one affects this part of the body, this affects the next part of the body, and this affects the next part of the body. That was an astonishing observation.
It was astonishing because it seemed too simple. Nobody else thought single genes were powerful enough to control something as complex as the structure of the body. Skeptics argued that Lewis's idea was guesswork. Of course, he had never seen the genes, because the techniques to do so didn't exist.
From the 1920s to the 1970s, it was not possible to physically isolate any specific gene. That opportunity first became available, fortunately for me, at the time that I was a student.
Levine enlisted his friend and fellow scientist, Bill McGinnis. The first gene they went after had an unusual name, Antennapedia, which means antenna leg. The gene was thought to control the growth of legs. When the gene misfired, flies grew legs in the wrong place. In normal flies, legs grow from the midsection, the area called the thorax. So Levine and McGinnis decided to hunt for the gene in the thorax of a normal embryo.
We wanted to light up the gene, and it was very painstaking work. And there was this moment when we saw that the gene was turned on in a band in the middle of a very early embryo. This had never been seen before.
The Antennapedia gene was acting like a master switch, turning on the segment of the embryo that would become the thorax. The implications were mind-boggling. If single genes like Antennapedia could define whole segments of an animal, these genes were acting like architects of the body. And if one of these genes turned on in the wrong place, striking changes to the body could result. It seemed that Levine and McGinnis had uncovered the genes responsible for the evolution of bodies. What about other animals? Did they use the same mechanism to build their bodies? An answer would come from Switzerland. In 1994, Walter Gehring of the University of Basel isolated the gene that triggered the growth of eyes in fruit flies. The gene was called eyeless because flies without it developed with no eyes. Gehring knew of a gene in mice that worked in the same way. He wondered, were the two genes the same?
And this question we tested by taking the mouse gene and putting it into fruit flies to see whether flies can understand the message of the mouse.
Gehring replaced a fly's gene for eyes with the mouse gene.
And to everybody's surprise, the mouse gene works perfectly well and can induce a compound eye in the fruit fly.
The fruit fly grew normal fruit fly eyes using a gene from a mouse. Not only did the two creatures use the same mechanism, they used the same gene. This was the mechanism behind extra wings, legs sprouting from heads, and Bateson's deformed animals. The century-long search was complete. The genetic engine of the body's evolution turned out to be a tiny handful of powerful genes.
So what this means is in some ways, some sense, evolution is a simpler process than we first thought. When you think about all of the diversity of forms out there, we first believed that this would involve all sorts of novel creations, starting from scratch, again and again and again. We now understand that no, that evolution works with packets of information and uses them in new and different ways and new and different combinations without necessarily having to invent anything fundamentally new, but new combinations.
Suddenly, the commonality of form among animals was understood. Animals resembled each other because they all used the same set of genes to build their bodies. A set of genes inherited from a common ancestor that lived long ago.
And what we see now among all the animals are just variations on a body plan that existed 500 million years ago.
And there's only one inescapable conclusion you can draw from that, which is if all of these branches have these genes, then you have to go to the base of that, which is the last common ancestor of all animals, and you deduce it must have had these genes. So the whole radiation of animals, the whole spring of animal diversity has been fed by essentially the same set of genes.
Ed Lewis shared the Nobel Prize in 1995 for the discovery of the universal set of genes that builds the bodies of animals. Most bipedal hominids went extinct, but one branch went on to evolve larger brains. That branch eventually led to modern humans. Our closest living relative is the chimpanzee.
Stand up.
We didn't evolve
nice.
from chimps, but we do share a recent common ancestor.
Can you walk over here?
That's why our DNA is nearly identical to theirs.
One of the great misconceptions that most people have is that once our ancestors stood up, it was almost inevitable that we would be here today, that the egocentric species, Homo sapiens, would evolve in this manner. But what we see is that evolution has worked the same way with us as it has with every single organism on this planet. We're here through a series of chance coincidences, specific adaptations, chosen opportunities. So I think that when we look at our own origins, we see that it is extraordinary that humans are here to look back and ponder their past.